I did. Yes. Honestly, all right, let's go ahead and get started. So, we've got 4 weeks left, which means if you're keeping a paper chain count of how many lectures there's left, I was informed this morning there's 9. So we're almost done. So what does that mean? Then I just want to take a few minutes to kind of overview what we've got coming up over the next few weeks, just to kind of keep you guys up to date with everything. Due dates on everything are largely posted through the end of the semester. Okay, so this week there was a homework assignment today, there's one for Wednesday, there's one for Friday. Next week for Monday is actually when you and your group of 3 or 4 total will start to work on your individual lectures. And these, I'll spend some more time talking about them on that day. But the information is up for it now, so that means I've got under this Unit 4, I've got a separate section that has the rubric and the expectations and what this presentation actually is. You could probably break it into 3 basic parts. 1, a 4 to 5 minute lecture. 2, standing up here and answering questions for 2 to 3 minutes. So these would be questions from the class. You can bet I'll have one if the class doesn't, okay? And then 3, coming up with at least two questions that will kind of be like the review questions for your section, because these actually will become part of that final exam, okay? So what you present will become part of the final. Danica. Yes, you do need to answer them as well, and that's kind of all in the description. So we'll talk about this some more on Monday, okay? But those questions and the presentation would both be due the Monday after Thanksgiving, okay? For next Monday, by the end of class, you need to have a topic picked out, and this is a topic of something that interests you that we have not talked much about this semester, okay? And you also need to have one objective. So if it's simply know the signs and symptoms of some illness, there's your objective, okay? If you hand it in before class, this is my incentive to getting you guys to work ahead of time on this so that in class is spent working on these presentations. I'll throw in an extra credit point. So it's a 2-point assignment. It's a 2-point assignment to get the objective and topic to me by the end of Monday next week class. I'll give you 3 points if you hand it in to me before class. The reason why I'm doing this is because I'd like to see you guys working. During that time, I'd like to see you actually going into the literature and finding information instead of coming up with what the idea is. Okay? So that's what you'll have for Monday. Obviously, what it will be is I've got already up in that section, I've got a couple of separate like drop boxes. It'll take you to a wiki that is for you and your group to post those questions, or to post the topic and objective for me. Okay, and then I have the same sort of thing for the questions, and we'll take a walk through this on Monday next week. Haley.
So you have to approve of what we're gonna,
Honestly, not really. I'm really not going to say it. The only time I might disapprove of it would be like, let's say your topic is antibiotics. We've spent, 4 or 5 weeks talking about antibiotics, so that would be one that I'd say like, no, that's not a good topic. But largely, this is a chance for you guys to look at what you want to, Haley, so feel free to explore with it, okay? All right, so that's the first part, or that's one thing I wanted to make sure I mentioned. The other is, don't forget, Tuesday doesn't affect you guys next week, but there is no class, there is no lab on Wednesday next week, okay? Because the cutoff time for everything is 12 o'clock on Wednesday when you're out of here for break, and since class falls after that, and since one of the lab sections falls after that, your only obligation next week for this class is Monday afternoon. That's it. Okay? You may want to take some time between then and Wednesday to take a look at your results from the experiments you do on Friday, but that's kind of at your own pace. So yeah, 3 lectures on immunology and pathogenicity. Today we'll cover innate immune responses. Wednesday we'll cover adaptive immune responses. And then on Friday we'll talk about pathogenicity factors or virulence factors. So in other words, proteins, etc., that give microorganisms their disease-causing abilities. We'll talk about some of those various factors that have nothing to do necessarily with immunology. Okay. Now we've got student presentations for 3 class periods, and then we've got 4 more lectures. 2 of those are going to be vaccines. Our last 2 will be kind of like an overview of types of vaccines, and then our last lecture is the controversy of vaccines. And we've kind of got 2 in the middle that I set aside for epidemiology. But to be honest, I'm not sure if I want to do that. So what I would like to do, probably sometime next week, is after we get done with this set of lectures, for us to decide as a class, do we want to continue talking about immunology, and we can go a little bit into, we can talk about a couple of other features that we're not going to be covering here, or do we want to talk about epidemiology, which is just how we identify like the point source of a disease. To be honest, that section is kind of like alphabet soup in some ways. It's a lot of terminology. Okay, so I'm just kind of giving you a heads up of that. So next week we'll revisit that real quick to see where do you guys want to go with that and with those lectures. Any questions? All right, so today what we're going to do is we've got 3 main objectives. We're going to talk about or describe the process of phagocytosis and its contributions to immunity. We'll talk about the contributions of inflammation to immunity, and then how do microbes overcome these various innate immune responses. And one of the ways to maybe set this up a little bit is just to remind, these are not terms that I've asked you to remember, and I'm not sure why I didn't put them in there, but innate versus adaptive immune response. Okay, innate is more generalized. What gets recognized are patterns that are common among all microorganisms, or among classes of microorganisms. The adaptive is going to recognize something very specific about one microorganism. So, what is a common feature among all gram-negative bacteria? Outer membrane, which has what on the outer face of that outer membrane? LPS, porin. LPS would probably be the one that I would go with here, because LPS is a molecular pattern. It's a pathogen-associated molecular pattern, or a PAM. Those are the types of features that the innate immune system recognizes, are things that are common among all within a class or a group of microbes. We'll take a look at a few of these later on. To begin, let's start with this. Okay, so I'm going to give you 5 steps of phagocytosis. Okay, so we've got destroys pathogen attachment, maturation or maturing phagosome, phagosome forms, phagolysosome forms. These are 5 steps, and what I'd like you to do is 1, put them into order, in terms of from first to the end of phagocytosis, and then secondly, I want you to come up with a couple of sentences to generally describe what's happening in each of these steps. Okay? Feel free to use your notes. Feel free to use the slides. Take 5 minutes to walk through this with at least one partner. All right, so here kind of, in my mind, this is the order. And note, the terminology that I gave you for those steps is not the exact same that's up here, and that's okay. The goal is not to memorize the names of the steps, but more to know what happens during these steps. Okay? So when we say attachment, when we refer to attachment here in phagocytosis, oftentimes we're talking about the attachment between the foreign material and the phagocyte, or the microorganism that is, or sorry, the cell, immune cell that is performing this phagocytosis. And in today's context, let's just talk about macrophages and neutrophils. Okay? What has to happen for that attachment to occur? What actually interacts with each other? Receptor on what, Abby?
On the phagocyte.
Good. On the phagocyte. With what? What does it interact with?
Foreign particles.
Good. And specifically, more specifically, what? What's the term that we brought up already? Has a fun acronym.
Yeah.
PAMP. Thank you. So a PAMP, a pathogen-associated molecular pattern, interacts with the receptor on the cell surface. These receptors that we have on the surface, there are some of them shown here. So like this is showing one known as C3, one known as lectin. I just want you to know the term PRRs, or the term here means, oh my gosh, why am I blanking right now? Sorry. Go to the next slide. Thank you. Pattern recognition receptor. Awesome job. All right. And TLRs are a specific subtype of these. The TLRs are the ones that are probably we know the most about. One of the first to have been recognized. They recognize features like the toll-like receptors will recognize lipopolysaccharide, teichoic acid on gram-positives. Some of them even bind to extracellular DNA and RNA. And they usually don't get, they will usually find them inside of the cell, but that's a whole different story. So the goal of these PRRs is to bind to the pathogen. Okay? Once that interaction takes place, then we get what? What's the next step here? Phagosome forms. What is a phagosome?
It's like a vesicle that takes in the foreign particles.
Perfect. Just kind of like what this image is showing. So we've got this interaction at the cell surface. What happens is the phagocyte, or that macrophage, begins to undergo some sort of change on the cell surface. It responds to that interaction. That response is it begins to kind of bowl inward, and eventually we form an endocytic vesicle. Okay? And that endocytic vesicle in this case is just called a phagosome. So phagosome forms, microbe is inside of it. Phagosome matures, which includes what? What was that? Drop in pH is the major feature here. And then eventually it fuses with the lysosome. What is a lysosome? Or what is the function of a lysosome?
Doesn't it carry garbage?
It does oftentimes, yeah. It's basically meant as a way to break down material inside of the cell. So oftentimes what we find with the lysosome is it contains in it proteases, in other words, enzymes that break down proteins. Oftentimes we'll see a low pH. Oftentimes we will also see reactive oxygen species. Okay, so there's kind of three features occurring here. The lysosome produces nitric oxide, reactive oxygen species. We've talked about the importance of reactive oxygen species and how some microorganisms have catalase to combat with that. Well, the amount of catalase they're producing oftentimes is not good enough to account for the amount of reactive oxygen species that are being produced within the phagolysosome. So oftentimes the cell ends up becoming oxidized, the proteins become oxidized, the nucleic acids become oxidized. That leads to damage to the cell, oftentimes to death. We've got enzymes that are breaking down the cell from the outside in, and we've got a low pH condition as well. All three which would contribute typically to the death of the microorganism. Okay? At this point, what happens? That's what we cover on Wednesday. Okay. Option 1 is it gets discarded. Option 2, parts of that pathogen get presented on the surface of the cell. And that's how these innate immune cells, like a macrophage, will signal to the adaptive immune response that, look, there's something foreign inside the body, we've got to kill it now. Okay, so that's how we get an overlap between the innate and adaptive immune response. Danica.
What else is involved in like the maturing besides the drop in pH?
That's the biggest one. There are other protein changes that occur in there too. So like some of the proteins that we might find in like a phagosome are actually pH dependent. Ones that as the pH drops, they become active. And so we might see some changes like that. But kind of the major one I want you guys to know is about the pH drop. Yeah. Okay. Let's take a look at a couple of ways by which microorganisms overcome this. Okay, so we'll take a look at, I've got more than just 2 examples, but we'll take a look at just a couple today. Let's start with this one. So you may recall, when we were talking about transformation a few weeks ago, we talked about an experiment performed by Frederick Griffith, and I drew this. Maybe the most memorable thing of that was this really crappy mouse drawing, right? And the mouse gets infected with smooth streptococci, and the mouse, well, I'm sorry, that's what happened. The mouse dies, right? And then the mouse gets infected with rough. And what happens? When infected with the rough, what happens? That's if you get rough and smooth infecting at the same time. But if we just infect with rough, it lives, right? All right, you still have to, oh God. All right, so this is kind of a follow-up experiment. All right, so in this follow-up experiment, what was performed, this was 2010. They ended up taking either rough or smooth streptococci, strep pneumoniae, and they put them into a petri dish that contained neutrophils. And so there's no recruitment or anything, like recruitment of neutrophils or anything in this example. The neutrophils are already there. They allowed the rough or smooth to incubate with the neutrophils for 30 minutes. And then did plate counts at time 0 and 30 minutes afterwards, calculate the CFU. And then what this y-axis here represents is a ratio. How many were there after incubation and how many were there before incubation? Okay. I should probably include a couple of more bars that might make this a little bit easier to understand. Yep. So why, so the basic question is why, what would explain those results? That there are more smooth after this 30-minute incubation than there are rough cells when incubated with the neutrophils. It does not have anything to ribosomes. But that's a good guess. That is a good guess, given the context of the terms.
Wait, is the first, like, original graph material, that's with neutrophils present?
Yep, so these would be with neutrophils present. These would be with no neutrophils around at all. So this is just saying what you would normally see after 30 minutes.
So we're trying to figure out why the black and white result happens.
Yes. C. Do we need to get up and do jumping jacks? You guys look way out of it right now. Just being a passive observer.
I think that exam gave me a bacterial virus.
I don't think that could be true.
I think so.
Unless I laced one of them.
I have lung after that exam.
No. That's not possible.
I have pneumonia. That's a super infection.
Alright. What's the difference between the rough and smooth? Anybody recall? What was that page? The smooth make a capsule, the rough don't, right? The rough lack the gene that allows them to make a capsule. Go ahead,
I
Zach.
already got it.
Okay, go ahead.
Smooth got the capsule, so it has the receptors on the outside. The rough doesn't have the receptors. Or vice versa.
Let's try that again.
So the rough, they have the receptors, so then the neutrophils can phagocytose.
Okay,
The rough.
good. And why then are the smooth with the capsule not phagocytized?
They must attach to that instead of the receptors.
Yeah, I mean, in essence, what the capsule is doing here is it's kind of like a cloaking device in some ways. So the capsule, remember, we've talked about that its role is it's a sugar coat. We've said before that the capsule will prevent phagocytosis from occurring. In essence, that capsule is so thick here that it's much longer or carries out much further past the surface of the cell than any of those receptors do. So it basically prevents those PRRs from ever attaching to a PAMP in the first place, or one of those pathogen-associated molecular patterns. Exactly, that's what's happening. So what this is saying here is that one of the ways, but if we're going back to that third objective, how do microorganisms overcome, or what contributes to their pathogenicity? Here's one of those mechanisms. Presence of a capsule avoids destruction of those cells. It avoids their immune detection. Okay? Let's take a look at one more. Any questions on this one, though, before we move on? Allie?
So the, like, smooth and rough cells, whatever.
Yeah.
Do they insert, like, the cells of the mice? Like, is that what he's talking about? Like, the receptors are on the
Yeah.
cells?
Can I draw a picture and maybe this will help a little bit? So the rough and the smooth look the same. Nor if you're just looking at the cell, they're identical. Okay. They will both have teichoic acid coming out of the surface of them. That teichoic acid is also a PAMP, and the PRRs could attach to those. But what the smooth have in addition is that they've got this thick capsule coat, which kind of almost like hides those teichoic acid. Or the teichoic acid are kind of embedded in it. And so because of that, the PRRs are kind of like not recognizing that the teichoic acid is there in the case of the smooth cells, whereas those rough, since the teichoic acid is exposed, you can get a macrophage or a neutrophil to attach and then phagocytize it.
So these are, like, outside the cells, and then the receptors attach to them and pull them in.
Exactly.
Okay.
Exactly.
That's
So these would be out. These would be kind of floating around. You get attachment from the neutrophil. Now you get engulfment or phagocytosis into the cell. Exactly. Does that help? Yeah, and I guess I should have explained that. These CFU, these would be cells that are kind of still floating around in the media that they're incubating in. Yeah, thanks for asking that. Any other questions on this one? All right, let's take a look at one more. We'll go to Mycobacterium. What's one key thing that distinguish Mycobacterium from all other bacteria? They're acid fast, which means they've got what? They've got mycolic acid. Yeah, always do this. All right. So all Mycobacterium have mycolic acid. Keep that in mind as we take a look at this one. We're going to compare two conditions. We're going to compare wild-type cells to ones that are unable to produce mycolic acid. So we'll just call them, we'll just say no mycolic. And we're going to take a look at what is the external number of cells and what is the internal number of cells. Let's assume again that what's happening here is we've got these two different populations of Mycobacterium. We take a petri dish of neutrophils, add the Mycobacterium to the neutrophils, and then over 4 hours watch how many of the original population are left. So extracellularly, what we're going to find is that these cells actually get internalized pretty quickly. In other words, phagocytosis, at least the initial steps, the attachment, the engulfment do occur. And then what we find in the wild type cells as they get brought in, find that they actually accumulate. Okay. The mutants, though, accumulate and then disappear. So the question becomes, why? What could explain this difference between those that contain mycolic acid and those that don't contain mycolic acid? Okay. So this is saying that they both get internalized equally, but that there's something happening inside of the cell that accounts for the difference. What step would you pick out? What step of phagocytosis would you pick out? Killing them. And that's exactly what we observe. So the effect here wouldn't be something about the activity of the neutrophil per se. The only difference here is just something on the bacterial cell. Those without mycolic acid die. Those with mycolic acid survive. Where do we think those that are surviving are actually found in these neutrophils? If we could like peer into a neutrophil and then pick out the location of Mycobacterium, where do you think they would be located? They're located in the phagolysosomes, exactly. So what we find with Mycobacterium, and particularly the mycolic acid, what's the consistency of mycolic acid? Sticky. What's another term that we use? Waxy. Okay. If you pour water onto wax, what happens to that water? It beads up. It doesn't like permeate through that wax. It's the same thing that we observe here. The hydrogen ions, the reactive oxygen species, the nitric oxide, the enzymes, none of them can permeate that waxy mycolic acid. And so what we find is that Mycobacterium are actually incredibly resistant not to the initial stages of phagocytosis. The mycolic acid is actually a PAMP. But they are incredibly resistant to the destruction. Okay. You may recall that we talked about a couple of different antibiotics, ethambutol and isoniazid, and we said the target of those is the mycolic acid on these cells. This condition here, this no mycolic acid condition, this is what we actually observe when we add those antibiotics. that there's an increase in the ability to destroy mycobacterium by macrophages that are found within the lungs when we're treating that infection with isambutol or ethambutol or isoniazid as well. Basically, those antibiotics are giving our immune system a chance to catch up and a chance to destroy the microorganism. So 2 examples that we've taken a look at. Both are ones that the mechanism by which they inhibit the phagocytosis differs, but the end result is the same. The microorganism lives. Okay? We'll take a look at some more of these over the next couple of class periods and looking at how do microorganisms overcome either the immune response or what do they do to cause disease. Okay? What makes them pathogenic? To finish up today, what I want to do is I want to go back to inflammation. And I want to just kind of review the steps of inflammation for a couple of minutes. So let's start with this. 4 words: edema, vasodilation, margination, and diapedesis. When do we observe these events, inflammation, and what do they mean? Or how would you describe these events? Okay. Let's go for 4 minutes, and then we'll spend the last few discussing what's happening in inflammation. Describe, and then when are they occurring in the process? Like which might, if you're thinking about the process of inflammation as like a linear step-by-step process, what would we see first? What would we see towards the end?
Yeah, like that's the actual flow of the plasma. Yeah. Okay.
So, yeah, the plasma like through the
So in between like yeah, in between the epithelial cells.
I like the name of margination is like across both surrounding areas.
Maybe that should be a final example. Which is your favorite stack of inflammation? All right.
Oh, before they leave.
yeah.
Which
Wait, no, that's
MARGINATION. What? Vasodilation?
You always have that, no.
They're like
Yeah.
Hey, one thing to point out real quick. Do not confuse neutrophil with neutrophile.
Yes.
Okay? They are different things. Just wanted to point that out. Because I can't tell you how many times when we were talking about neurophiles that I would see the word neutrophil, and now I'm seeing the exact opposite. So.
I know. This is the first one that was. Yeah. About that.
Chemotaxis and diabetes, like, are they the same?
Kinda. I mean, chemotaxis is just describing the movement of the organism toward a
Okay. Right.
in the case of macrophages and neutrophils, margination and diabetes are two parts of that chemotaxis.
Okay, that makes
Yeah,
sense.
that's a great question.
That was definitely.
So the macrophages are what? Yeah, it moves out of the blood vessels.
We don't need to know.
All right, so let's just take a look at the broad overview here again. This is incredibly complex, and that's to be said about most of immunology. So do let me know if it feels like it's kind of spiraling out of control. It's very easy for it to do so. But we're just going to kind of think about this in the broad steps. So we've got some macrophages. This is an example that you'll see in every textbook for inflammation, but the main point is that there are some macrophages that are resident in tissue. So there are some lying right underneath the epidermis so that if you get a cut or an abrasion, they're there ready to respond right away. We don't call them the first responders. That's a term that we say for neutrophils that are being recruited to that site, but it's as if those macrophages are already on scene. The terminology for these resident macrophages, in all intents and purposes, it sucks, okay? And I was talking to Brenda about this earlier because you'll hear terms like Kupffer cells, or you'll hear alveolar macrophages or resident macrophages. They're all the same thing, okay? They are the cells that are there to begin with. And these macrophages, their job is, as soon as pathogens come in, they're ready to kind of sense and taste what's coming into the body at any given point. So here we've got a phagocytosis event, and then what does that macrophage do? Once it's sensed that this is foreign material, not only does it phagocytize, but what else does it do? It secretes the cytokines. And cytokines, in broad terms, are regulators of the immune response. Some enhance the immune response, some repress it, some will activate specific cell types. But overall, they regulate that immune response. Some are chemoattractants, chemokines. What do the chemokines attract? Neutrophils would be one type, okay? And let's just focus on that. Neutrophils and macrophages would be recruited to these sites of infection. And neutrophils, most abundant of the white blood cells in our system, also the quickest to make its way to that site of injury after it has first been sensed. So the cytokines have a couple of different effects. One is they bind, the cytokines can actually bind to these epithelial cells that make up the walls of the vessel. And what happens? They loosen. They start to space apart a little bit from each other. That allows for fluid to move into, or plasma to move into that tissue there. That often is the swelling that we observe with inflammation. That's what? What term do we give to that?
Edema.
That's edema. What else happens? What dilates? The veins, the blood vessels. Blood vessels dilate, vasodilation, increasing flow of blood cells through there, also resulting in drop of blood pressure. So if somebody goes into shock, one possible reason for it is inflammation is kind of out of control. Okay. And that vasodilation, that edema, also we will find neutrophils rolling along, and then what happens?
They start crawling.
Why do they start crawling?
I can tell you.
They hit cytokine or chemokine. They basically sense the cytokine or chemokine, and the analogy I've used in the past is like if you have watched a bloodhound pick up a scent before, right? It might be like looking around, and all of a sudden it's like, it stops and then it starts moving in the direction of where that attractant is. That's how these neutrophils respond, is that they're kind of just rolling around and then all of a sudden chemokine comes into, I can't snap, sorry, really crappy snap, okay? And then they start to sense that chemokine and start crawling along the vessels towards higher and higher concentrations of these chemical attractants. And at some point, they're going to find the highest concentration of it in the blood vessel, and then what has to happen for them to continue moving towards even more chemokines? They got to squeeze in between these cells, and that is what process? That's the diapedesis. So we find neutrophils will do this first. About an hour later, we'll find the macrophages following behind. In both cases, those neutrophils and the macrophages, their job is to eat, to chop up, or to basically consume everything that is extracellular, particularly foreign material that will bind to the PRRs, phagocytize it, break it down. In the case of neutrophils, neutrophils end kind of like a supernova, where they end up basically exploding all of their internal content, forming these nets, neutrophil extracellular traps. And these nets will trap bacteria, and they become actually part of like what we would consider to be the clotting and the scabs that form at a site of injury. But also, we've got macrophages that start taking pieces of those pathogens and presenting them on the surface and trying to get T cells and B cells involved in this process as well. That's how innate connects with adaptive immunity. Okay? So on Wednesday, what we'll spend the time talking about is how do T cells and B cells actually become activated? What processes of this presentation activate them? And then finally, what is their response? What do the T cells do? What do the B cells do? And how does that feed back to actually improve the innate immune response even more? Allie.
Do macrophages come from the capillaries?
Some macrophages will come from the capillaries. So we do have some that are actually kind of roaming through the blood vessels. There are also many strategically placed as kind of like there in case some injury occurs. Yeah, yeah, or like, you know, tonsils even. Yeah. All right, have a great afternoon. Spleen to filter blood everywhere. It's kind of ridiculous.